Hi, so we're learning about life science and cell biology in a nutshell. We were learning about membranes of cells and what makes them up, which is phospholipid molecules, as well as integrated various kinds of lipids, glycolipids, and um, specialized molecules such as proteins bound to lipids, etc. These all serve to make the membrane specific and help in its function. So lipoproteins, lipo um, polysaccharides. So different kinds of molecules are in the membrane. There's transport ATPases, channels such as porin that help with molecules getting across the membranes. So normally in eukaryote cells, you have just one single bilayer. A bilayer contains two rows of phospholipid molecules, along with all the other integrated components of the membrane, such as cholesterol molecules and protein molecules, like integral proteins, which serve as channels and different receptors and um, ion channels, etc. Molecules serving as for cell signaling. So also phospholipids come in four different types. So phospholipids we learned are these four different types. Basically, they all have the same glycerol backbone as well as a phosphate head group, which is on the polar end of the molecule, and they each have one of four kinds of head groups, alcohol type head groups. And on the nonpolar half of the molecule, there are two fatty acid tails. Okay, so so phosphatidylamine, serine, and choline look identical except for their head groups. Sphingomyelin has a slightly different backbone with nitrogen and one carbon replacing and another carbon replacing three of the oxygens that are in the other three kinds of phospholipids. So the head groups determine specificity of the membrane, its various functions, and attracting different proteins and enzymes for different functions. In their function, phosphatidylcholine and sphingomyelin are usually in the outer membrane of outer monolayer of bilayers. So the choline and serine are those two on the right, far right, in the outer membrane. Phosphatidylcholine and sphingomyelin are usually in the outer membrane. Phosphatidylserine and phosphatidylethylamine are usually in the inner membrane. Of inner monolayer of bilayers. So the first two are a little bit smaller in their head groups, while the second two in the outer are a little larger, bulkier molecules. The cholesterol molecule is distributed about evenly in both monolayers. So the inner membrane is more negative. So the cytosolic face of the membrane is more negatively charged. And a lot of enzymes that are important, such as enzyme protein kinase, are activated in response to various signals and require negatively charged phospholipids such as phosphatidyl serine for their activity. So the lipid head groups are important. They sometimes must first be modified so that protein binding sites can be created at a particular time and place. Phosphatidyl inositol, for instance, is a minor phospholipid concentrated in the cytosolic monolayer of cell membranes. So on the inner side of the bilayer, a variety of lipid kinases can add phosphate groups at distinct positions in the inositol ring. The phosphorylated inositol phospholipids then act as binding sites that recruit specific proteins from the cytosol to the membrane, so closer to the membrane. An important example of a lipid kinase is phosphatidyl inositol kinase, which is activated in response to extracellular signals, helps to recruit specific intracellular signaling proteins to the cytosolic face of the plasma membrane. So membrane phospholipids have an important role in the function of cell signaling. Extracellular signals activate enzymes such as PI3 kinase, phosphatidylinositol kinase, which phosphorylates inositol phospholipids in the plasma membrane. Various intracellular signaling molecules bind to these phosphorylated lipids, so from inside the cell, and are recruited to the membrane where they interact and help relay the signal into the cell from the outer extracellular space. These signals from outside the cell activate phospholipases that cleave phospholipids, the lipases. The lipid fragments act as signaling molecules, relaying signals into the cell. So the sites where different classes of phospholipases cleave phospholipids are shown here, where there's cleavage occurring. Phospholipases cleave ester bonds. So ester bonds are the oxygen, carbon, and oxygen double bonds, etc. So where you have two oxygens, one double, one singly bound, that's an ester bond. That's where these lipases function, cleaving at these phosphoester bonds or ester bonds. They can be a carbon or a phosphorus atom in the middle, and doubly bound oxygen and singly bound oxygen on either side, which makes it an ester bond. What did I do? Okay. So extracellular signal activated receptor protein. You have the kinase relaying signal, docked intracellular signaling protein, phosphorylated Phospholipid inositol.